This is a summary of everything we talked about in our cell reproduction unit. So first, to discuss why we reproduce in the first place. So number one, in a multicellular organism, remember, we started as a single cell. So in order for us to grow to the billions of cells we have now, cell reproduction needs to occur. Secondly, many cells have a limited lifespan. For example, white blood cells live 60 days or red blood cells live 120 days, and so these cells need to be replaced as they die off. Or if you get cut, cells need to be repaired or replaced in the case of damage. And finally, to make more organisms, either by asexual reproduction, where one organism divides and makes another organism, like in bacteria, or by sexual reproduction, where gametes are made and then fused together. This is the way we make more organisms. The important thing to know about cell reproduction is that the only real thing that's being passed on in cell reproduction is the DNA. So we need to start with a discussion of DNA, what it is, how it's stored, and how it's passed on. One of the characteristics of living things is that all living things have DNA, and they pass this DNA on. This is basically the directions for all the proteins that we make. Now, the way our DNA is stored differs between prokaryotes and eukaryotes. Prokaryotes, which are our bacteria, store their DNA as one circular molecule. That looks like this. It's still a double helix, if we were to imagine it. This is sort of a double helix. And then this would be our bacteria. There'd be no nucleus, no major proteins associated with it. It would be one linear chromosome. And the genome of that bacterium would be basically the DNA that's contained in that single chromosome. In eukaryotic organisms, we have multiple chromosomes. In humans, we've talked about in class that our diploid number, our body cells, contain 46 chromosomes. So this is our package of DNA. So if we were to take a single cell from me that contained all 46 chromosomes, that would basically represent all the directions in my genome. So you can think of a genome as a full set of directions for an organism. The smallest unit of a genome would be, in us, one body cell that contains all 46 chromosomes. So when we talk about the organization of DNA in a eukaryotic organism like us, our DNA is found in the nucleus. We usually think of the chromosomes as we see them in a picture as these X-shaped things, and we'll get to that. But when the cell's not dividing, we don't see chromosomes. We see the cell, we see the nucleus, and then we see some kind of a mass in there. And that mass is the chromatin. So chromatin is actually a mix of both DNA and protein. Now the point of the protein is that it helps the DNA to remain organized, because DNA is long and stringy and it takes up a huge amount of space. So if we look at DNA, DNA is made of nucleotides, and a nucleotide consists of a sugar, a phosphate, and a base. The important part here is the phosphate. So phosphate looks like this, and it has these negative charges on it. What that does is it makes DNA have an overall negative charge. It then is attracted to these proteins called histones that have the opposite. They have a positive charge. And so what's going to happen is, when we don't see the DNA, when the cell is not dividing, the DNA is being copied. It's doing its job, which is to make proteins. So it unwinds, sort of like opening a book. And so certain portions of the DNA at a particular time might be coiled up, if these are histones, might be coiled up around these histones, but then other portions of the DNA might be unwound because maybe that's DNA that's being copied at that time. So if we imagine it, these histones sort of like little spools and the DNA is wrapped around them, and maybe this DNA opens up because this portion needs to be read to make a particular protein in the cell, but other parts are staying all coiled up. So that's what chromatin really is. It's DNA and these histones that basically control when the DNA is being read and when the DNA is just being stored. It allows the DNA to take up 50,000 times less space than it would if it was completely untangled. So it helps to keep it organized, sort of like if you had a whole bunch of thread and you wrapped it around little spools, so that it didn't get tangled up. So the diagrams here sort of give you an overview of what we mean when we talk about the DNA being packaged. So DNA is a double helix, but if we were to take that double helix of DNA, if we look at this picture on the right here, we can see that the yellow circles represent histones, and those histones actually then coil up themselves, and what we end up with when a chromosome is fully coiled is this whole fuzzy looking thing that's actually 50,000 times longer if you were to untwirl that DNA into one long strand. Now, as far as chromosomes go, you need to know the basic structure of a chromosome. And I'm not going to go over all of it here. This is from your homework. But I will make a quick comment. The centromere we've talked about in our notes. That's this little connector part here. But also, the shorter arm, or when they're organized, the top arm, is always called P, and the bottom arm is called Q. You also should look up what the function of a telomere is. You'll notice that telomeres are always found at the end, and they're going to be important actually a couple chapters from now. Now, one important thing you need to understand about a chromosome is that the amount of DNA in a cell, if it's never ever going to divide again, is not as much as the amount of DNA in a cell when it's going to divide again. In other words, the cell has a cell cycle. And at the beginning, when a cell is first born, if you were to coil up the DNA, even though it wouldn't normally be coiled up, it would look like this. It would be one linear stick with the centromere in the middle. Only if the cell is planning to divide will that stick make a copy of itself, and now you would have, here's again the centromere, and these two sides are called sister chromatids, and they are exactly identical to one another. The only reason they're there, we now have twice the DNA, is so that later on, when this cell goes to divide, this can be split in half. One will go one way, one will go the other way, and each new cell will end up with basically one of these sticks. Here's another diagram showing the same thing. Here's what your chromosomes would look like prior to S, if this was a cell in G1. Which we'll come back to. And then after S, in G2, this is the amount of DNA you would have. Notice how this side is just an identical copy. It's completely unneeded. The only reason it's hanging around there is so that when this cell divides, each new cell will get a copy of the information. It's sort of like if you made a photocopy of your notes. You don't need a second copy of your notes, but maybe you were planning to share them with somebody. Now you would have a copy, and you could give that photocopy to your friend. So what's going to happen when the cell divides is that you have these duplicated chromosomes, and they've got two chromatids. Now what will happen is the chromatids will separate. Each one will go to its own cell. These will not be called chromatids anymore. Right now, this is called a chromosome with two chromatids, but in the end, when these separate, each of these is a chromosome on its own. I guess you could say it's consisting of one chromatid, but you don't usually use the word chromatid at all if it's just a single one. So this vocabulary is really, really important. It's going to come back a few times when we get to genetics as well. Make sure you know the difference between a somatic cell and a gamete. Somatic cells are body cells, and in us, in animals, our somatic cells are always diploid. That means we have two copies of each chromosome. Our gametes are haploid. They have only one copy of each chromosome. This does not mean that a diploid cell has this and a haploid cell has this. This is not two copies of a chromosome at the top here. What this actually is one chromosome that happens to have two chromatids. So what we would mean by two copies in one copy would be, for example, if this was a cell with a 2n or diploid number of 2, then a haploid cell would have an n number of 1. So in us, our 2n or diploid number is 46. That's what our body cells have. But when we go to reproduce, we use meiosis to cut this in half, and so our n number, which is found only in a sperm or an egg cell, is 23. And again, the purpose for that is now when fertilization occurs, this will restore the number back to 46. Otherwise, if there was no meiosis, if we never cut the chromosome number in half, every time a sperm fused with an egg, the chromosome number in the offspring would double, and again and again and again, which wouldn't work. So this is a picture called a karyotype, and again we see centromeres and sister chromatids, and in a karyotype there's a lot of things that we can tell actually. The chromosomes have specific bands and they're lined up in pairs. So what this is showing is a diploid cell, and there are 2 copies of pair number 1 and 2 copies of pair number 2 and 2 copies of pair number 3. If you were to do a karyotype, let's just say of a sperm cell, you would only see 1 copy of chromosome number 1, 1 copy of chromosome number 2, etc. Another thing we can tell from a karyotype is whether, if we're talking about humans, whether it's a male or a female. Since this one has an X and a Y, which you can recognize because the Y is much smaller, this would be a male. In human females, there would be 2 X's and no Y. So for the cell cycle, what you need to know is this whole thing here is interphase. This is basically when the cell's doing all of its important jobs. Whatever that cell's job is, it's going to be doing it during interphase. If a cell is never ever going to divide, it'll actually kind of split off from here into something called G0. A brain cell would be a good example of a cell that usually stays in G0. But if a cell is going to divide, what's going to happen is G1, the cell's doing its normal jobs. S, this is where the chromosomes are going to go from sticks to X's, so that when mitosis comes along, that extra copy can be given to a new cell. Mitosis is this whole section here. We don't have to know the parts of it for the purposes of our class. You do need to know that technically it's not cell division, it's nuclear division. So in order for a cell to actually split in 2, it actually has to go through mitosis and then also this extra step called cytokinesis. So you end up with a cell that ends up splitting, and then you actually do end up with 2 cells. If cytokinesis does not occur, what you would end up with is 1 cell, and it would basically have 2 nuclei. And a good example of a cell in your body that does this is the muscle cell. So this is sort of an overview of mitosis in detail. However, you don't have to know the individual phases. So I'm going to give you a little sketch on the right. Let's just pretend like we have a cell with 4 chromosomes. So it has a 2n number of 4. Now, before S, remember those 4 chromosomes would have been sticks, like this. We can also pretend like they have little centromeres in the middle of them, like that. But after S, those chromosomes wouldn't be sticks anymore. Now each of those 4 would be X's. Alright, so what's going to happen is this cell is going to go into mitosis, and again, I'm drawing out the chromosomes. Remember, technically, during this time, you couldn't see them. They would be in a mass called chromatin, which would be a mix of the DNA and the protein, and some areas would be coiled up, etc. Alright, so what's the most important thing that happens during mitosis? Well, those chromosomes, the 4 of them, would line up in a straight line down the middle, the equator, and they would be attached to these fibers called spindle fibers. Looks like this. You have these little centrioles at the ends. And then what's going to happen is the cell, or the chromosomes, are going to get pulled apart. So we're going to assume that this cell is also going to undergo cytokinesis, so I'm showing you the cell splitting at the same time. So here's our 4 chromosomes, and here's our 4 on the other side. And what's happening is they are getting pulled towards opposite sides. So the X's literally split in half. In the end, what we're going to end up with are 2 cells, and they are going to each have 4 chromosomes. They're going to look like sticks. Here they are. And notice, if we go back to our original cell in G1, it looked exactly like this. So this would make exact copies. One difference between plant cells and animal cells is that animal cells in cytokinesis go through cleavage. And what actually happens is microfilaments, and you need to remember those. Microfilaments were one of the types of cytoskeletal elements, and microfilaments can contract. So when they contract, they're made of actin, and this literally causes almost, they call it like a little noose that actually splits these into daughter cells. On the flip side, what happens with plant cells is a special thing called a cell plate forms, and now we end up with our two cells that are split by a new cell wall. Later, some plant hormones will cause these cells to stretch, so you will end up with growth, but the point is there is no pinching apart. In prokaryotes, we have what's called binary fission. I'm not going to go into a lot of detail, but basically that DNA would make a second copy of itself, and then you'd end up with the cell splitting in two, and you'd have each cell with a loop of the DNA. No nucleus, no spindle fibers, just very, very simple. It actually takes a lot less time. I think this can happen in like 20 minutes, whereas mitosis, let's say in an onion cell, ends up taking like 13 hours or something. So this is a really simple, fast process in prokaryotes. They also have a lot less DNA. Alright, as far as the cell cycle being regulated, we don't want cells to reproduce out of control. So what ends up happening is there are these checkpoints throughout the cell cycle. You don't have to understand this completely, but here's the deal. You've got enzymes called cyclodependent kinases. You should recognize that. It ends in ase, so it's an enzyme. And proteins called cyclins have to hook up with the kinases, and when both of these are present, it tells the cell to move on to the next stage. So there has to be a signal, or a cell would go into G0, like brain cells. Or if a cell's damaged, like in the liver, they can go from G0 and go back into the cell cycle. My understanding is that the kinases are always there. The enzymes are there. But the cyclins are not always there, and so it's the production of the cyclins that then causes the activation and everything moves on. And there's apparently different types of cyclins and CDKs. It's not like there's one cyclin-dependent kinase. There's different types found in different kinds of cells, but it's very, very controlled. And this diagram just shows an example. Like we said, there's checkpoints. The issue is that if a checkpoint gets ignored, you could have a cell that's reproducing uncontrolled. And when that happens, that's what cancer is. Cancer is when a cell kind of goes rogue and reproduces uncontrolled. Maybe it makes its own growth factors, it ignores growth factors. We don't completely understand. Most cancer is actually random. It's not necessarily genetic. But we do know that normal cells show two things that cancer cells don't. One is density-dependent inhibition, and that should actually be dependent. And what that means is that when one cell it grows and it starts touching another cell, that the cells send signals to one another and basically tell each other not to divide. The other thing that cells normally show is called anchorage dependence, meaning that they should be anchored to something. If a cell breaks off and floats away and ends up in the wrong location, it shouldn't reproduce because it's going to be in the wrong spot. There shouldn't be cyclins telling it to reproduce if it's not located in the correct area. So cancer cells basically ignore both of these. They ignore density-dependent inhibition, they are not anchorage dependent, and they just keep making copies of themselves. This diagram just illustrates it, that these cells will only grow if they are anchored to the bottom, and when it fills up, they stop growing. But if you were to take some cells out, they would start growing again to refill the space. In cancer, we would have something like this, where the cells just keep reproducing, even though the entire bottom of the dish is covered. They're ignoring the fact that there's no signals there telling them to divide. Like I said, maybe they're making their own signals telling them and cells around them to divide when they shouldn't be. The only thing I would comment on from this quick slide would be it's called transformation if a cell becomes cancerous. We know there's things called carcinogens that tend to make cells transform. Normally, our immune system looks for damaged cells and destroys them. Tumors, that's what we call that mass of tissues. If they're staying in one spot, they're called benign. If they're moving or metastasizing, moving to other areas and invading other spaces, then they're called malignant.